Hello friends, my name is Shani and today we'll talk about protein. In the previous lecture we talked about different kind of biomolecules that are present inside of our body, and among them protein is one of the most important kind of biomolecule that is present. Protein is a homopolymer or it might be a heteropolymer of different monomer units that are known as amino acids. So protein is a homopolymer or it might be a heteropolymer whose monomer units are called amino acids. So more than one amino acids. Amino acids are basically of 20 different types inside of a body. So 20 different amino acids that are present in a body, they make up a single protein moiety. So you see that there will be so much different combinations of these amino acids that will make a single protein. So that there might be more than 1,000 amino acids in a protein. These amino acids, as the name suggests, there is amino group that is the most crucial part of these amino acids that will contain nitrogen atom. We'll talk about the structure of amino acids, the roles of amino acids, the types of amino acids, and how they form a protein, what reactions are involved in making a protein. So we'll talk about those all steps in this lecture. First, we'll talk about the structure of amino acids and what are amino acids. Amino acids. Amino acids has a central carbon component. You can call amino acid as a substituted methyl group where all the hydrogen atoms are substituted by different kind of moieties or groups. As we'll see in a common structure of amino acids, there will be one amino group, that is NH3 plus. So there will be one amino group. There will be one carboxyl group, COO minus. There will be one hydrogen atom. So you can see the three valencies has been occupied by different groups: amino group, carboxyl group, hydrogen group, and there will be one R group. R group. As you can see, these three groups, amino, carboxyl and hydrogen group, these three groups will be common in any amino acid, but the different types of amino acid that we have talked about, 20 different types of amino acid, that will be differentiated on the basis of this R group. This R group may be a simple moiety like hydrogen, like a methyl group, or it might be a complex structure such as some aromatic ring, and according to that 20 different types of amino acids has been classified in human body or in any other animal. So this R group can be a simple hydrogen group, so it will be known as the glycine amino acid. I'm just giving a few examples of amino acids which are important. Out of 20, I'll mention a few. If the R group is the CH3 methyl group, then it will be known as alanine. If this amino group will be some complex moiety, like it will be CH3OH, as it contains hydroxyl group in it, so it will be serine. If it contains any other aromatic group, like if it contains this kind of benzene ring which is attached to the hydroxyl group, so it will be known as phenylalanine. Here it will be CH3 that will be joined with this, so CH3 aromatic ring, and then it will be linked to the OH, and here it will be linked with the carbon atom. So as the R moiety, it determines different type of amino acids which will be present. So this is the basic structure of the amino acid. So central carbon will be occupied by 4 different groups: amino group, carboxyl group, hydrogen atom, and one R group. That will be different types of R group in a single amino acid, in a different amino acids, that will be known as different R groups. So this was the basic structure of the amino acids. Now we'll talk about how or what kind of amino acids are there and how they will be involved in the formation of the peptide group. So this was the structure. Now we'll talk about, there are 2 kinds of amino acids basically, on the basis of the requirement by the body or their synthesis by the body, on the basis of synthesis by the body. So if amino acids can be synthesized by the body, few of them amino acids, like we have talked about, out of these 20, those amino acids that can be synthesized by our own body, those are known as non-essential amino acid. And those amino acids that cannot be synthesized by our body, that means they only have to be obtained by diet. So those amino acids are known as essential amino acids. As the name suggests, your body cannot synthesize it. You have to obtain it by the diet. So they are essential. By diet you mean you can have it by different kind of protein-rich diets like eggs, milk, or any other diet. So essential amino acids are those that can not be synthesized by body, and non-essential, so they can readily be synthesized in a body, so they are not necessarily to be required to be obtained from diet. So they are synthesized in the body. So these 2 kinds of amino acids are generally present in nature. So now non-essential, essential, and now amino acids, as I told you, have different kind of groups in them. So I'll draw a structure once again. As we have seen that there will be central C component, there will be one amino group, and there will be one carboxyl group, there will be a hydrogen atom, and there will be an R group that we talked about. So this amino acid, as you can see, has a positive charge as well as it has a negative charge, which will eventually neutralize each other, giving it negative or net zero charge. If this COO group is occupied by one hydrogen atom, so you will see that there will be C, one amino group, one hydrogen atom, one R moiety. So now this COO will be occupied by this H. So this amino acid form has a net positive charge. And when this amino group will lose one hydrogen atom, it will have net negative charge. So we'll see there will be a central atom C, there will be a hydrogen group, there will be this COO minus, and there will be the R moiety. But if this amino group loses one hydrogen, then it will lose its positive charge. So it will be converted into NH2 amino group. So this amino acid has a net negative charge. So as you can see, depending on the hydrogen ion concentration, or you can say pH of the surrounding medium, the amino acid can readily be present in various kind of charged conditions. It might be positively charged, it might be negatively charged, or it might be neutral charged. And this neutrally charged form of the amino ion is known as Zwitter ion. It is known as Zwitter ionic form of the amino acid, and this form is very important in forming your protein, that is the basic monomer of your body. So Zwitter ionic form will be, it is the most prevalent form of the amino acid in the body. Now we'll talk about how protein is formed by different amino acids. We have talked about amino acids, their types, their structures. As for now, we don't have to necessarily remember all the 20 amino acid names, but if for just extra information, if amino acids has to be classified on the basis of the R group and their nature, they might be classified in different categories. One is your non-polar amino acid. One is your non-polar amino acid that is not charged. Non-polar, not charged. One amino acid is polar amino acid. With positively charged, and one is polar amino acid with net negative charge. So there will be different kind of amino acids on the basis of the nature of the R group. If it is non-polar and if it is not charged, like as in the case of glycine, we talked about that there will be simple H group, right? And in the case of alanine, there will be simple CH3 group. So it has net negative charge, because this negative charge and this positive charge, they will neutralize each other. So they will be non-polar because there is no polar moiety at all, and it will be net negatively charged. So it will be non-charged, non-polar amino acid, polar amino acids with some positive charge. They include the examples of lysine, arginine, because those amino acids have a positively charged R group. For example, in the case of lysine, this R group is CH2, 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 and finally NH3 plus. So you will see there will be a positively charged amino group in their structure. So as the same way, negatively charged polar amino acids will have net negatively charged R group. As in the example of glutamic acid and aspartic acid, they will have net negatively charged. That means they will have a COO group, COO minus group in their R moiety. So this was the classification on the basis of the nature of the R group. On the basis of the nature of the R group. Now we'll talk about how the protein is formed, how the different amino acids, they combine to each other by forming a protein. So as we have seen that there are an amino group and a carboxyl group in a single amino acid. So when two amino acids has to be combined, the amino group of one amino acid and the carboxyl group of other amino acid, they react with each other with a reaction where a water molecule is released. So we'll see that suppose this is one amino acid, glycine. In the case of glycine, there will be this NH3 positive here. This is H. R moiety will be H. So here the COO minus. Suppose the other amino acid that it has to combine with is phenylalanine. So phenylalanine contains C. Here it will be H. again here it will be NH3 plus, it will be COO minus, and it will have CH2, and then there is this phenyl ring and OH group. So this is phenylalanine. Now when this carboxyl group and this amino group has to react with each other, they will release an H2O, a water molecule. So 2H, 1H from here, and if it will be present in the form of COOH, here it will be NH2, it will be COOH, so it will be NH2. So one hydrogen from here and H and O from here are released. So it will eventually lose one hydrogen from here and oxygen and hydrogen from here, OH, thereby forming H2O. So they will be bound to each other by this bond CO.NH, and so these two amino acids will be linked to each other, and this bond where one hydrogen molecule is released when two amino acids bind to each other is known as your peptide bond. This is your peptide bond. As the same way, this is one peptide bond, another peptide bond will form here with this amino acid and the upcoming next amino acid, the next peptide bond, and so on, and it will form your protein molecules, the whole protein molecules that might contain more than 1,000 or more amino acids. So this was the basic process of the formation of the peptide bond between two amino acids and the formation of a protein. Now I'd like to introduce different kind of proteins that exist inside a body. What are the basic proteins that are present inside of a body? So you might have heard about collagen. Collagen is a major protein that is present in our body. That is, you can say it is present in extracellular matrix. It is a component of extracellular matrix, which makes your skin, your skin cells. Okay, then most of the meat that you eat from different animals, that is mostly collagen. So it is present in your skin, in your muscle. So that is collagen. The next kind of protein that we commonly observe is trypsin. Trypsin is a kind of enzyme. We'll talk about enzyme in a different video. So trypsin is a kind of enzyme. Then you can say insulin. Insulin is a hormone. Then antibodies. Antibodies are also a class of proteins that are required for defense reactions of your body. They are generally fighters of your body. So in immunity. So this was different kind of proteins. There are more receptors. Receptors are generally present on the cell membrane that are majorly responsible for receiving different signals from outside of the body. So receptors. Then there will be different proteins like GLUT4, which is required for the glucose transport across the cell. So different kind of proteins, collagen, trypsin, which is an enzyme. Insulin is a kind of hormone that insulin is responsible generally for reducing the glucose level in your blood. Then there are antibodies. So these are different kind of proteins that are present in our body. Now we'll talk about the structure of the protein. We have talked about how the protein is formed. Now we'll talk about how the protein fold is, or how the protein makes different kind of structures. So protein structure. So there are generally 4 kind of protein structures. One is primary protein structure, secondary protein structure, tertiary protein structure, and quaternary protein structure. As we have seen that proteins are made up of amino acids, so the sequence of amino acids which forms the proteins, so that sequence is known as the primary structure of the protein. So primary structure is nothing but the sequence of the amino acids, of the different amino acids that form that protein. Like for example, if a protein is made up of glycine, phenylalanine, valine, glutamic acid, aspartic acid, so on. So this linear structure that shows the amino acids that are present in the protein, they are known as the primary structure of the protein. Now, secondary structure, when this amino acid or this primary structure has folds on itself. It has fold on itself at a certain places. Like this is a linear structure of amino acid. So this is the primary structure. Now, when it folds like this, so it will form helical structures, or it can form different sheets. So these turns that are introduced in the primary structure, they will form the secondary structure. So secondary structure, some amino acids has turns on the peptide or polypeptide chain. As this primary sequence contains more than amino acid and it contains more than one peptide bond, so it is known as polypeptide. Poly means more than one and peptides mean peptide bonds. So it will contain more than one peptides. So if this polypeptide chain turns on itself, I mean it has turns on it certain particular places like here, 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 so they will form the secondary structure. So it chain will be like having certain kind of helical structures. That will be known as secondary structures. Generally there are two kinds of secondary structures. You can say this is a kind of helical, a spiral structure, so it is helix, and one more structure is sheet. Some sheets are formed due to the turns and different folding of the proteins. Now, if we talk about tertiary structures, when this peptide chain or this polypeptide is forming or it is revolving around itself and it forms a kind of structure that is three-dimensional. So 3 stands for three-dimensional structure. So it will be a 3D projection. So the polypeptide chain will fold on itself and it will again fold on itself and it will form a kind of three-dimensional structure that will be known as its tertiary structures. Now, if we talk about quaternary structures in primary, secondary and tertiary structures, there was only one polypeptide chain that was involved in forming those structures. But if we talk about quaternary structures, there will be more than one polypeptide chains that will be interacting with each other and that will form a kind of loop or a kind of structure that will be known as quaternary structures. So what you have to remember is in quaternary structure there will be more than one polypeptide chain. That the example of protein which shows quaternary structure is your hemoglobin, that is present inside your RBC, red blood cells, that carry oxygen in your body. So hemoglobin has 4 polypeptide chains that will be linked by different bonds in between the polypeptide chains. So these are the structures of the protein. Primary structure, a simple sequence of amino acid, a linear structure. Secondary amino acid, when this simple sequence of amino acid, linear structure, folds at a certain residues and it will form a kind of helix or it will form a kind of sheet. So they will be known as the secondary structure. tertiary structure, it contains the sequence that will fold upon itself and it will form a kind of three-dimensional structure. The quaternary structure will contain more than one polypeptide chain. So these was all about the structure of proteins and how different kind of amino acids make up about the protein. So in the next class, we'll talk about different biomolecules like carbohydrates and nucleic acids. Thank you for the video. If you like the video, please like, share and subscribe our channel.